Dear viewers, so up till now we have been discussing the different techniques in spectroscopic methods. In the earlier episode, we were discussing the inductively coupled plasma. So today we will be continuing with the same technique, and we will also discuss another technique, which is atomic fluorescence spectroscopy. So let us first of all continue with ICP, that is inductively coupled plasma. Let us go through the lecture plan of the today's lecture. Today we will be discussing the ICP source, its methodology, transformation into the sample, instrumentation, integrated techniques, applications of ICP. Then we will start with the next technique, that is fluorescence spectroscopy. We will study its introduction, its principle, fluorescence and chemical structure, factors affecting fluorescence, quenching, instrumentation, fluorimeter and general layout of a fluorimeter, then its application. So this is all about the lecture plan. Now let us start with the lecture. So earlier we were discussing what is ICP, its general introduction and principle was discussed in the earlier episode. Today we will see the ICP source. It consists of 3 concentric silica quartz tubes, each of which is open at the top. Liquid sample is nebulized into an aerosol, and a fine mist of the sample in the form of droplets is introduced into the ICP. The nebulizer argon stream that carries the sample passes through the central tube, which is also called injector. The excitation is provided by 2 or 3 turns of a metallic induction tube through which a radio frequency current flows. The second gas flow of argon at a rate of 10 to 15 liter per minute maintains the plasma. The plasma gas flows in a helical pattern which provides stability and helps to isolate thermally the outside quartz tube. The plasma is initiated by a spark from a Tesla coil probe and is thereafter self-sustained. So this is the figure which shows the ICP source. Here we have 3 concentric circles. This is the central one, where we have the central flow, then we have the intermediate flow from the middle one, and from the outermost we have the outer flow. This is the active zone, which is shown by this lined part. This is in short called AZ. Then we have the induction coils, these are shown by the dots. Then we have the central channel, the middle part, and the recombining zone, that is RJ, this is the outer part. So this is the diagram of ICP source. Coming on to the methodology. Argon is circulated throughout the coaxial tube. On applying radio frequency field, argon gets charged and produces electrons and charged argon ions. A spark is applied at the mouth of the coaxial tube to produce the plasma. Liquid sample to be analyzed is introduced into the plasma to puncture it and encapsulate the sample at a very high temperature within it. Transformations into the sample. A radio frequency field causes ionization and formation of plasma, and then the sample flow punches a hole in the plasma. The sample of the aerosol is carried into the center of the plasma. The sample undergoes various transformations like desolvation, vaporization, atomization, ionization and excitation. During excitation, the valence electrons are excited to higher energy state and a very high temperature in the plasma is obtained. The gas temperature in plasma is around 6273 Kelvin. So such a high temperature is obtained. It helps to eliminate chemical interferences. This high temperature, it helps to eliminate the chemical interferences, and the emission from plasma is polychromatic. Polychromatic means that many different wavelengths are present in the this emission. Separation is done by a monochromator because we have a polychromatic emission. So, and we need only one wavelength, to the separation of the required wavelength is done by using the monochromator. Detection is done by a photomultiplier tube. The instrumentation part, two types of instruments are used in plasma emission analysis. First is the simultaneous spectrometer and the second is the sequential element spectrometer. In the case of simultaneous spectrometer, the radiation from the plasma enters from a single slit and is then dispersed from a concave reflection grating. The component wavelength reaches a series of exit slits which isolate the selected emission lines for specific elements. The light from each slit is detected to fall on the cathode of a photomultiplier tube and one for each spectral line is isolated. The resulting voltage is converted into concentration of the elements. In sequential element spectrometer, the plasma is located in the upper center of the spectrometer above the nebulizer. The light from the plasma passes through a lens onto a mirror and then it passes to monochromator and the grating. Light then travels through the second monochromator and grating, which improves its resolution. Solid samples are dissolved or digested in liquid. Usually an acidic solution is used. The sample solution is then sprayed into the core of the inductively coupled argon plasma, which can reach temperatures of approximately 8000 degrees Celsius. At such high temperature, all analyte species are atomized. That means they are converted into atoms. They are then ionized and thermally excited, and then they can be detected and quantified with either an emission spectrometer, that is ICP OES, or a mass spectrometer, ICP MS. So, these are the integration of two techniques, or we can say the combination of two different techniques. So, coming on to the sample types and preparation, ICP techniques are robust and suitable methods of trace metal analysis applicable to a wide array of sample types. Metals, plastics, proteins, soils, ceramics and many other can be successfully analyzed using ICP methods. We have the integrated techniques. Integrated techniques are the techniques which uses the combination of two techniques. Like we have ICP MS, this is inductively coupled plasma combined with mass spectroscopy. Then we also have ICP AES, this is inductively coupled plasma combined with atomic emission spectroscopy, or ICP OES, this is emission spectroscopy again. Coming on to the application of ICP AES in the determination of metals in wine. So, the integrated technique ICP AES is used in the determination of metals in wine. Also, it is used in the determination of arsenic in food and in the determination of trace elements bound to proteins. It is also used for analysis of trace elements in soil, and it is often used in forensic science to ascertain the origin of soil sample found at the crime scene or on victim's body. ICP AES is used for motor oil analysis. Analyzing used motor oil reveals a great deal about how the engine is operating. ICP AES is also used during the production of motor oils and other lubricant oils for quality control and compliance with production and industry specifications. ICP OES is widely used in mineral processing to provide the data on grades of various streams for the construction of mass balances. Coming on to the applications of ICP MS, it is used in medical and forensic field, specifically in toxicology, heavy metal poisoning, metabolic concerns and even hepatological issues. Depending on the specific parameters unique to each patient diagnostic plan, sample collected for analysis can range from whole blood, urine, plasma, serum to even pegged red blood cells. In the environmental field, the ICP-MS finds its application. Such applications include water testing for municipalities or private individuals, all the way to the soil, water and other material analysis for industrial purposes. In the forensic field, glass ICP-MS is popular for glass analysis. Trace elements on glass can be detected using the LA-ICP-MS, where LA is laser ablation. ICP is again inductively coupled plasma and MS is the mass spectroscopy. The trace elements from the glass can be used to match a sample found at the crime scene to a suspect. ICP-MS is also used in the geochemistry for radiometric dating, in which it is used to analyze relative abundance of different isotopes. In the pharmaceutical chemistry, ICP-MS is used for detecting inorganic impurities in pharmaceuticals and their ingredients. For example, ICP-MS may be combined with size exclusion chromatography and quantitative preparative native continuous polyacrylamide gel electrophoresis for identifying and quantifying native metal cofactor containing protein in biofluids. So, this was all about the ICP. Coming on to the next technique, which is atomic fluorescence spectroscopy. So, let us first of all look at the introduction of this technique. Luminescence. The word luminescence basically is the emission of light by a substance. It occurs when an electron returns to the ground state from the excited state and loses excess energy as a photon. It is of two types, fluorescence and phosphorescence. Fluorescence occurs when a molecule absorbs light at one wavelength and re-emits the light at a longer wavelength. Fluorometric analysis is very sensitive and widely used method for quantitative analysis. Fluorescence starts immediately after the absorption of light and stops as soon as the incident light is cut off. So, this is the property of fluorescence. The second type of luminescence is phosphorescence. It occurs when light radiation is incident on certain substances and they emit light continuously even after the incident light is cut off. So, this is the difference between fluorescence and phosphorescence. Fluorescence stops as soon as the incident light is cut off, while phosphorescence continue a little later after the incident light is cut off. Possible electronic spin state of a molecule. So, this diagram shows the spin state of a molecule. There are two types of spin state, singlet state and triplet state. Suppose a molecule has two electrons. So, in the ground state, both the electrons are paired in the opposite spin. One is in the clockwise spin, so and the other will be in the anti-clockwise spin. And the spin of an electron is considered to be plus half or minus half. In singlet state, the two electrons, they are not paired and they have opposite spins. That means, if one is having a spin of plus half, the other would be having a spin of minus half. In triplet state, again the electrons are not paired, but they are in the same spin. That is, both will have spin either plus half or minus half. So, how this singlet and triplet is calculated? We have the formula for spin multiplicity 2s plus 1, where s is the spin of the electron. In case of singlet state, we have spin 0 because spin of one electron is plus half and the spin of another electron is minus half. So, the total spin is 0, plus half minus half makes 0. So, 2s plus 1 makes 1. Since the spin multiplicity is 1, therefore, it is a singlet state. In the second case, we have two electrons, both are in the same spin. So, their spin adds up. So, half plus half makes 1. So, 2s plus 1 makes 3. Therefore, this is the triplet state. So, this is how we have singlet state and triplet state. Fluorescence and phosphorescence. So, this is the diagram by which we can see what is fluorescence. We are basically concerned with fluorescence. This is the ground state S naught. There are different excited state. The electron jumps from the ground state to a higher level. Now, from the higher level, the electron, it comes back to one of the excited state by losing some energy, and thereafter it comes back to the ground state. Now, when it comes back to the ground state, the energy difference is low because it has already emitted some energy in coming from one of the excited state to a lower excited state. So, now the light it emits, it is of longer wavelength, and this is the fluorescence. Each molecules contain a series of closely spaced energy levels. Absorption of a quantum of light energy by a molecule causes the transition of an electron from the singlet ground state to one of the number of possible vibrational levels of its first singlet state. Once the molecule is in the excited state, it returns to its original state in several ways. One of them is the fluorescence process. The vibrational equilibrium before fluorescence result in some loss of the excitation energy. Therefore, the emitted fluorescence is of less energy and longer wavelength than the excitation light. Only the molecules capable of absorbing UV or visible radiation can show the phenomena of fluorescence. Greater is the absorbance of a molecule, the more intense is its luminescence. Hence, the molecule with conjugate double bond or with pi bond can be studied using fluorescence, because in these cases the absorbance is more and the intense luminescence is obtained. Fluorescence and chemical structure. Fluorescence is most common in compounds containing aromatic functional group with low energy. Fusion of heterocyclic nucleus to benzene ring increases fluorescence. Example, we have these three structures. The first one is quinoline, the second is isoquinoline, and the third is indole. In all these three cases, there is a fusion of heterocyclic nucleus to the benzene, and therefore, in all these three cases we obtain an increased fluorescence. Substitution on benzene ring shifts the wavelength of absorbance maxima and corresponding changes in fluorescence peak. Fluorescence decreases with increasing atomic number of the halogen. That means, we have halogens fluorine, chlorine, bromine, iodine. So, if we compare the fluorescence of iodine with chlorine or fluorine, we will found since this iodine is of higher atomic number, it will show less fluorescence. Fluorescence is inhibited by the substitution of carboxylic acid or carboxylic group on the aromatic ring. That means, if we have a carboxylic group on the aromatic ring, it will not show fluorescence. Fluorescence is favored in molecules with structural rigidity. That means, the molecules which are structurally rigid, those shows more fluorescence. Organic chelating agents complex with metal ion increases fluorescence. Like for example, we have this is fluorine and this is biphenyl. So, these are the chelating agents, that means, these form chelates with metal or these form rings with metal ion. So, these shows more fluorescence or these increases the fluorescence. Coming on to the factors which affect fluorescence. First is the nature of molecule. Fluorescence is only shown by the molecules which absorbs UV visible radiation. Greater is the absorbency of the molecule, the more intense is the fluorescence. Second factor is the nature of substituent. Electron donating groups enhances fluorescence. Example, OH NH2, these donates the electron to the ring and increases the fluorescence. On the other hand, electron withdrawing groups like COOH, NO2 etc., these decreases or destroys the fluorescence. The third factor is the concentration. Fluorescence is directly proportional to concentration. For a more concentrated solution, it will show more fluorescence, and a dilute solution will show less fluorescence. Next factor is dissolved oxygen. Dissolved oxygen or DO reduces the emission intensity of fluorescent solutions. This reduces the fluorescence intensity because of two reasons. The first is by direct oxidation of the fluorescent substances to non-fluorescent, and second is by quenching of the fluorescence. Next factor is pH. pH of a solution has a significant effect on fluorescence. For example, we have aniline. In case of aniline, neutral and alkaline solutions of aniline shows fluorescence in visible region, whereas the acidic solution shows fluorescence in the ultraviolet region. The next factor is temperature. Increase in temperature decreases the fluorescence. So, temperature is inversely proportional to fluorescence. And next factor is viscosity. Decrease in viscosity decreases the fluorescence. So, we can say that fluorescence is directly proportional to viscosity. The next factor is intensity of incident light. Increase in the intensity of incident light on sample increases fluorescence intensity. Intensity of light depends upon number 1, light emitted from lamp, and number 2, excitation monochromatous, number 3, excitation slit width. Decrease in fluorescence intensity due to specific effect of constituent of the solution. This is called quenching. So, quenching is basically decrease in the fluorescence intensity. It is of various type. First is self quenching, then we have chemical quenching, we have static quenching and collision quenching. Coming on to the self quenching. This is caused by excessive absorption of primary radiation by the solution, which is also called concentration quenching or inner filter effect. Deviation at high concentration is also due to the self quenching. As for example, we have in this graph, we have plotted fluorescence versus concentration of the fluorescing species. So, at low concentration we get a straight line, but at high concentration there is a deviation from the straight line. So, this is due to the self quenching. Chemical quenching. This quenching occurs due to actual change in the chemical nature of the fluorescent substance. This can be due to the change in pH, presence of dissolved oxygen, halides and heavy metals. Example, aniline gives fluorescence at pH between 5 to 13. At pH lower than 5 and greater than 13, it does not show any type of fluorescence. Next is static quenching. This occurs due to complex formation. Example, caffeine reduces the fluorescence of riboflavin by complex formation. So, in this case a complex is formed which reduces the fluorescence. This is called static quenching. Next type of quenching is the collision quenching. In collision quenching, fluorescence reduces due to collision. As the number of collision increases, quenching takes place. Coming on to the instrumentation part of fluorescence spectroscopy. The basic components of a fluorimeter and spectrofluorometer are number 1, source of light, 2, filters and monochromators, 3, sample cells, 4, detectors. So, these are the components. Now, coming on to the source of light. The source of light can be mercury arc lamp, xenon arc lamp, tungsten lamp, tunable dye laser. Mercury arc lamp. Mercury vapors at high pressure produce intense lines above 350 nanometers. Mercury vapors at low pressure gives additional radiation at 254 nanometers. Xenon arc lamp. It gives more intense radiation than mercury lamp. A continuous spectrum is obtained over the range between 250 to 600 nanometers. The next type of lamp is tungsten lamp. It is used if the excitation is to be done in the visible region. Next is tunable dye laser. Here pulse nitrogen laser as the primary source is used. Radiation in the range between 360 and 650 nanometer is produced. Coming on to the filters. These are optical filters and works on the principle of absorption of unwanted light and transmitting the required wavelength of the light. We have two types of filters, primary filters and secondary filters. Primary filters are the one which absorbs visible light and transmits the ultraviolet light, while the secondary filters are those which absorbs ultraviolet light and transmits the visible light. A monochromator converts a polychromatic light into monochromatic light. It is of two types, excitation monochromators and emission monochromators. Excitation monochromators are the one which isolates only the radiation which is absorbed by the molecule, and emission monochromators are the one which isolate only the radiation which is emitted by the molecule. Coming on to the sample cells, which are also called cuvette. It is used for holding liquid samples made up of quartz and can have cylindrical or rectangular shape. The path length is usually 1 centimeter. Path length means the width of the cuvette. It is usually 1 centimeter. Coming on to detectors. Detectors can be photovoltaic cell, phototube, photomultiplier tubes. Photomultiplier tube are most commonly used as these are the best and accurate. These are more sensitive because these can measure the weak intensity of light also. The principle employed is that the multiplication of photoelectrons by secondary emission of electrons takes place. A photocathode and series of anodes are used. Each cathode is maintained at 75 to 100 volts higher than the preceding one. An amplification of 10 to the power 6 is obtained. Next is the fluorimeter. It is of two types, single beam fluorimeter and a double beam fluorimeter. Single beam fluorimeter uses tungsten lamp as a source of light. Here primary filters absorb visible light and transmits UV lights. The emitted radiation are measured at 90 degree by secondary filters, and secondary filters absorb UV radiation and transmits the visible radiation. The advantage of using single beam fluorimeter is that these are simple in construction, easy to use and economical, and the disadvantage is that it is not possible to use reference solution and sample solution at the same time. Second disadvantage is the rapid scanning to obtain excitation and emission spectrum of the compound is not possible. So, a double beam fluorimeter is generally used. Here two incident beams of light source pass through primary filters separately and fall on either sample or reference solution. The emitted radiation from sample or reference pass separately through secondary filter. The advantage of using double beam fluorimeter is the sample and reference solution can be analyzed simultaneously, and the disadvantage is that rapid scanning is not possible due to the use of filters. This is the general layout of fluorimeter. Here we have power supply, then the source, then we have primary filter, then we have the sample cell, the slit, then comes the secondary filter, and after that the detector, and finally the data processor. Coming on to the applications of fluorescence spectroscopy. The first application is the determination of inorganic substances. Example, determination of ruthenium ions in presence of other metals. The second example is determination of aluminium third alloy. Determination of boron and steel by complex formed with benzoin is the other example. It is also used in nuclear research in field determination of uranium salts. It is also used as fluorescent indicators, mainly used in acid base titration. For example, eosine. Eosine is colorless in acidic solution while green in basic solution. Fluorescein, another indicator, it is colorless in acidic solution and green in basic solution. Next indicator is quinine sulfate, which is blue in acidic solution and violet in basic solution. Next is acridine, which is green in acidic solution and violet in basic solution. In organic analysis, qualitative and quantitative analysis of organic aromatic compounds present in cigarette smoke, air pollutants, automobile exhaust, etc. Liquid chromatography. Fluorescence is an important method of determining compounds as they appear at the end of chromatogram or capillary electrophoresis column in the determination of vitamin B1 and B12. So, these are the applications of fluorescence spectroscopy. Coming on to the summary. Today we have studied the remaining part of the inductively coupled plasma, which included the ICP source, methodology, transformations into the samples, instrumentation, the integrated techniques, application of ICP. So, these were all about the ICP. Then we started the next technique, which was atomic fluorescence spectroscopy. We studied its introduction, its principle, fluorescence and chemical structure, factors affecting fluorescence, quenching and the various types of quenching, instrumentations, fluorimeter, the two types, single beam and double beam, the general layout of fluorimeter and the applications of fluorimeter. So, this was all about today's lecture. In the next upcoming lecture, I will be taking the next topic, which is also the last topic of the spectroscopic methods, the electron spin resonance. Thank you.